a little bit about standards and some of the examples of how we're doing this in our database, which is the Comparative Toxicogenomics database. And like a lot of us here, we're interested in some aspect of how the environment affects human health via some things that happen in between, whether that's perturbing gene expression, protein expression, pathways. And a lot of these data exist. And this has been brought up numerous times, particularly in this session, about even though the data exists, is it really in a format that we can do something useful with? And a lot of it comes down to language. And some of you have seen this slide before, but these are just examples of the left-hand column. Those are all names for the same exact gene. On the right-hand side, those are all names for the same exact chemical. And although we have identifiers for these things, they are not always unique, as you heard in the last talk. So the problem with this is resolvable to a certain extent, and some of it is will, and some of it is just, oh, got a little problem there, not able to actually coalesce the community to have buy-in and use these standards in a way that would propagate the data in a more consistent way. So genes are probably our least big problem at the moment, and I'm sure a lot of you who are molecularly based have been gratified with a lot of the work that has gone on at NCBI, trying to resolve cross-species names, and we've gone from having PPAR gamma from what used to be many, many different names. So genes have come a long way. And those are sort of less problematic because these are entities that don't necessarily have hierarchical relationships to each other. And so the field of ontology development has really exploded in biology in recent decades, particularly in the last decade. And so I am not an ontologist, but I unfortunately, like probably a lot of you who are building resources, have had to get involved with ontology development or understanding on a level that maybe you didn't realize existed. And so ontologies are not just the terms, but mapping the relationships between those terms. And this becomes really important with a lot of the broader concepts that we're trying to model in biology. So not just a chemical interfering with a gene, but at what levels of biology are the consequences for that perturbation. So why are standards important? They're important for interpretation, and I'm going to show you just some real simple examples of each of these things for extrapolating data. And this is particularly important in toxicology, where we're not exposing, at least on purpose, human populations, but we're using model systems and trying to understand how those may influence or help us interpret what the risk might be to human populations. For curation, both manual, which is what my group does largely, but also for automated mechanisms that are clearly under development by lots of groups to try to increase the pace at which we can categorize and extract data. And then ultimately integration to get at these sort of broader concepts. So this is not a new problem. A lot of us say, well, nobody uses standards. We actually use standards all the time, right? At least most of us, maybe those who are not politicians anyway. We use words and we know what each other are talking about, right? Because we have definitions for these words. And with some exceptions of fact and fiction these days, there's fairly good consensus about that. So why we can't do this in biology is a little bit frustrating. So as I mentioned, genes and proteins, we're pretty good on these relative to where we were 10 or 15 years ago. And as you've heard from several people, we're not so great on chemicals, phenotypes, diseases. And so just as a small example, and a lot of the frustration for me, because we curate our own data from the literature, but we also incorporate several public data sets that you would think some of these groups were actually had a little more buy-in on some of these standards than they do. But for example, if you're interested in phenotypes that involve behavior, and you search by any of these phenotypic terms on the left, which arguably are related to one another, you come up with very, very different results in a disease database. And so this becomes really difficult to manage if you're trying to pull that in on a regular basis and integrate it with lots of other data that may be equally as messy as that. So in cases where we have better concordance between things like gene names, you can actually really augment your interpretation and understanding. So just as an example of genomic data, we have very little phenotypic data for human genes when it comes down to it. But if we look at what we have from other model organisms and we know what those orthologs are in human, we can actually build that phenotypic representation of genes from about 35-ish% up to about 75% if you believe in evolution and can make that leap from a zebrafish to a human and know you're talking about the same gene. So for extrapolation, this is similar to that example with the behavioral differences. So it depends on the communities that are generating these data too, and this becomes really important as we try to access them and pull in clinical data from clinical repositories and understand epidemiological data. So a clinician looking at Parkinson's disease may use very, very different terms to describe their patients than, say, somebody who is working in a laboratory trying to develop a mouse model for Parkinson's disease. And if you're looking at this from a computational perspective, it may be a lot harder to pull those things together and say, yeah, we may be talking about some very similar phenotypes. And so the manual side of this becomes very, very important because it's hard to see those similarities. And this is a really nice example, I think, from Melissa Hendel, who's out in Oregon and is really a pure ontologist, like somebody who gets super excited about ontologies. She's the one. And we need people like this because to take data from very, very different sources and figure out that, yes, in fact, we can use the zebrafish and look at aspects of swim bladder and know that maybe we're modeling something about lung function in mammals. Takes a lot of steps, right? And so you can't be looking necessarily at the really high-level anatomical features because we don't have swim bladders and they don't have lungs. But if you can look at very different levels, more granular phenotypic levels, you can maybe make those connections across these different kinds of organisms. And what's really important about doing this is that you have very standard ways of describing these different features and phenotypes in different systems. So for our database, which is CTD, we curate a number of different aspects of toxicology. So the goal of this database, for those of you that might not be familiar with it, is to bring together information about chemicals, molecular targets, phenotypes, diseases, and now we also have an exposure module. And we curate these data from the literature and try to bring this together to create sort of a broader view of what we can understand and predict about health outcomes with respect to exposure. And so the data we have in there is human all the way down to invertebrates. And we try to facilitate that cross-species look and do the kinds of things that I showed you in that previous figure from Melissa. So this is kind of a breakdown of the data that we curate. So we curate chemical gene interactions. We do this the best that we can with chemical representation, knowing that there are limitations that we heard about in the last talk. We also curate chemical disease and gene disease relationships, and I'm going to talk a little more about these last two because they're newer initiatives for our database. So one of them is an exposure module. So we got involved in this several years ago really before the whole explosion of the exposome discussion, but trying to figure out how we can bring together exposure data on a level that would be useful for the exposure science community. And then also, so we had been for many years curating sort of chemical gene disease relationships, but knowing that there's a gigantic leap between the genes and the disease, and they're actually, you know, cellular level, tissue level, phenotypic changes that may be occurring before that, that could help inform or predict diseases that come out of that initial exposure. So these are the ontologies and vocabularies that we use. Chemicals and anatomy are from MESH, and we know there are limitations to those. Genes, we use NCBI, Entree gene. Diseases, we kind of made our own mashup of OMIM, which has very, very granular genetic diseases, and the MESH disease combination, and that's all publicly available and has been adopted by a number of model organism databases rather than having sort of multiple things. So we manually mapped these OMIM, these granular OMIM diseases to higher, more descriptive diseases that were in MESH. For species, we use NCBI, and for phenotypes, we use a combination of things, and I'll show you more about what that looks like. And then for the exposure database, this was really my unfortunate foray into ontology development, but there really wasn't anything out there that would sort of encompass the complexity of exposure data, and so we embarked on what I naively thought was going to be sort of a quickie meeting of community people, and we would, you know, spit out this sort of structure, and it turned into this multi-year thing. And so the way that we approached that was to develop sort of a broad framework and then just start curating the data and populate it as we go, rather than another approach, which some people take, is, you know, sort of flesh it all out and then see if it accurately represents the data. So all of these things are publicly available in Medic. We have a paper describing exactly what we did, as well as with EXO. So if people are interested in that, there are papers on it, and I'm happy to talk to you more about it. With respect to manual curation, I'm glad I put this up. We initially, years ago, started with Excel spreadsheets and had all the problems associated that everybody else has had, but just to assure you, our data is not curated in Excel spreadsheets. We have web interfaces that interact with our database, and we have a shorthand way that our curators capture very, very specific types of concepts, and all of the data is mapped against the database, so we don't run into issues where cast numbers are dates, which we are familiar with that problem. So this is just to give you a little idea about the exposure ontology, or the exposure curation that we do. We have over 50 different categories, so this is really different from our much simpler chemical gene interaction information. This is sort of a spreadsheet view of that, but you can see the major concepts that we capture are stressors. These are typically chemicals based on the focus in our database. We have receptors, which are, say, the populations that are exposed. So a lot of these data are epidemiological studies. The exposure event, we have 17 different categories of data, and if I had one wish associated with this project, I would get all the epidemiologists in one room, lock the door, and not let them leave until they could come to some consensus about how to report their data, because trying to capture in a structured way all of the numerical and measurement data and statistics that they generate, it is wildly all over the place and very difficult to do in a standardized way, but I think we've come up with a solution. So I'll show you a little bit about what that looks like in the database in a second. As far as phenotypes, this is actually pretty complicated too, given the breadth of the kinds of models that we look at. So it's anything from in vitro to model systems. And again, trying to bridge that interim between an exposure and a disease, but capturing things that are in between. And this is a little bit of a simplified version of what we curate. So this is based on sort of a community-based approach for capturing phenotypes, and it uses a lot of different ontologies. So we capture a chemical, we capture, you know, a degree of change, we capture what the actual effect is, where it occurs, what kind of cells, what taxon. So the degree of information is quite specific. And then everything in our database is always grounded in a manuscript that's published. So what does all this mean? So we make sure that we capture everything using these control vocabularies. We also incorporate other data from other data sets that are publicly out there and integrate it in such a way that you're not just doing what you would do with a PubMed search and finding a paper, but you're actually finding a conglomeration of information that will give you a bigger picture of what's there. So I won't kill you with screenshots, but just as a little view of what I was just showing you, if you look at a particular compound such as cadmium, you have all these tabs that would show you information that we have curated or integrated relative to that chemical. For instance, diseases, because of the structures we use and the standards we use, that can be grouped hierarchically into sort of broader categories, or if you're interested in a particular category, you can look at that and see all of the very specific disorders, mental health disorders in this case, that are associated with cadmium. There's a little like bug flying around there. So we have relationships between chemicals and diseases. We also have relationships via genes. So this gives you some idea of what a potential mechanism might be that links an exposure to a chemical to a disease. We have computational tools where you can look at those 44 genes that would make a connection between Alzheimer's and cadmium and look at their connectivity. So I don't have time to show you all the tools we've developed, but a lot of this comes out of both our curated data as well as other data that we've integrated, and that can only happen through that use of standards. So for the exposure data, what you would see is this is just a quick view of, you would get a list of all the epi studies that looked at cadmium. You can get a quick view of who was studied in them. You can go to the references. If in cases, a lot of epi studies don't report the actual levels that are measured, some do. If they are reported, we provide those. And this is not trivial, I'll just tell you, to get all of that into the database, because a lot of these are, as has been mentioned, in PDF files, which is really frustrating. And in our last release of the exposure data, which was about a month ago, we had previously only been showing a portion of the data because we capture so much exposure data, and now you have the ability, you probably can't read that, but these are basically all of the categories of data we have for every study, so you can pick and choose what kind of data you actually want to look at. So, so that's for just a quick view of data you can actually get when you go to CTD, but other things that we're working on, and this you'll hear a lot more about this kind of approach in the next session. But again, for cadmium, we have all of this information. We have genes that are perturbed, we have phenotypes, we have diseases, exposures, and a lot of this sounds a lot like the data that people are trying to put together for adverse outcome pathways. And so we've been playing with some of that, and again, you'll hear more about this, but thinking about, can we build adverse outcome pathways computationally? And of course, these would need a lot more granular and manual looks, but you could potentially start building some of these at a high level. So we know what sorts of genes are perturbed, and we have connections between those chemicals and phenotypes, as well as genes and phenotypes. and then connections between these phenotypes and individual diseases. And all of this can be banked on publications, both from epidemiological literature as well as from model systems. And then finally, I'll just mention we're also working on a project with David Reif and his graduate student Marissa, who's back there, where we're integrating ToxCast and Tox21 data with CTD with the hope that we could take what is a large chemical space from ToxCast, much larger than what we have curated data for in CTD. However, there's very little phenotypic data and limited gene data. And so our goal is that by virtue of integrating these things, we can actually bring a broader context, biological context, to the ToxCast data. And then the flip side of that is that we can increase our chemical space in CTD with a lot of the screening work that's been done at the EPA and NTP. So I'm just going to end with, so I've just talked about a few very, very small aspects of standards, and we've been really careful about what kinds of standards we use and trying to be mindful of what other resources are using so that we can really leverage other data sets out there and integrate them. But I think there are still some remaining challenges, like really establishing consensus across which of these ontologies we want to use, and how to incentivize people to actually use them rather than to just go out and, you know, show their data in the way that they're most comfortable with if it's not really consistent with the community's opinions. And then finally, building tools that you can use to navigate these ontologies, because if you're not in this world and you're trying to play nice, it's not easy to figure it out. And the tools that are available for building them, I'm friends with a lot of the people that build these tools, and I told them I can't use them. They're impossible to use, and they know it. And a lot of it is, you know, really funding and support for these kinds of tools so that they can be done correctly. So there are a couple of resources out there where you can find standards, but there's a huge amount of redundancy across these things. And so there's a lot of work that needs to go into, I think, you know, both building, harmonizing, and incentivizing use. And I just want to point you to a paper that came out a few days ago from Melissa Handel and her friends and family, where they, I think, did a really, really nice job of distilling down, and these are conversations that have been going on for years, and I think this is a really nice paper where they point out, and it's not just for the average scientist, but it's also for resource builders, and they come up with a list of recommendations for how to approach using standardized language and how to reference that and make sure that that's being done well. And particularly this idea of using stable identifiers, which kind of seems like a no-brainer to a lot of us. And this particularly jumped out at me yesterday because our software developers have been, we incorporate data from a database that my software developer said, don't mention which one it is because they might be there and then you'll offend them. So they will, it will remain nameless. However, they have completely changed the way they structure their identifiers, which means we go in and do our monthly load, but now the identifiers aren't matching up with anything in our database, so we have to go back and change. So it's like that kind of thing is so avoidable, and I would love it if we could all get on the same page with that. And fortunately, we have an administration who's going to be really supportive of science and moving forward with this. So I think it's really up to us and the community to embrace this sort of non-sexy area of science and talk to each other more about how to talk about data. So I will stop there, and The CTD team is a small but live and really wonderful group who does the lion's share of the information capturing and everything that you see when you go to the database. And we have some great collaborators at NC State, and our funding is almost entirely from NIEHS. And we just got refunded, so you'll have to put up with me for at least a few more years. So I'd be happy to take any questions.